Hello, this is Kelly Eversole, the Executive Director of the International Alliance for Phytobiomes Research, and I'd like to welcome everyone to our webinar series today. We're fortunate to have Denise Manker with us from Bayer Crop Science, and before we get to her presentation, I'll give you a little bit of an introduction about the Phytobiomes Alliance. We are an international alliance and a nonprofit consortium of industry, academic, and governmental scientists. We have a number of sponsors, which does include Bayer, but our sponsors range from small startup companies to very large agricultural companies to research institutes to governmental research institutes as well. And because of our sponsors, we're able to support this webinar series. So just to remind everyone that phytobiomes is a complex system of plant-based agriculture. It's the whole idea that we have a plant in a particular place, in a particular biome, and it's surrounded with a specific environment that includes climate, it includes microorganisms, macroorganisms, other animals, other plants, and in most cases it's in soils, but clearly in other environments such as indoor agriculture, doesn't include soil always. But it's the whole concept of an entire system in that environment. And it's all influenced by management practices that are being done by either growers or ranchers or foresters in the case of forest systems. Our vision is that by 2050, all farmers will have the ability to use predictive and prescriptive analytics based on the geophysical and biological conditions of their specific field or specific site, and that they'll use that information to determine what's the best combination of crops, management practices, and inputs. Just want to remind everyone, we have a Phytobiomes conference in 2021. It'll be held in Denver in September, and we're trying to cover the full range of scientific topics. So I would encourage you to go to the website. It's pretty easy to find, just phytobiomesconference.org, and check it out. And we hope to be rolling out our new agenda within the next couple of months. So our next webinar will be on the 22nd of October, and we'll have a focus on the 4R nutrient stewardship program, which is a really interesting component of phytobiomes and a great way to move towards sustainability. To remind you, the webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channel within a couple of days. And the presentation will actually be followed by a Q&A session. And please use the Q&A panel if you have questions, and you can start putting those in as soon as the presentation begins, and then we'll ask the questions at the end. If you would like to chat with the panel or with the attendees, then you can do that through the chat panel. But do not put your questions in the chat panel because we don't always see those. You already have access to both my presentation and to Denise's presentation, so I would encourage you to go ahead and download those. So with that, I'd like to turn it over to Denise Manker. She's the Senior R&D Fellow for Biologics at Bayer Crop Science, and she's going to talk to us about the fascinating journey from discovery to product. So Denise, thank you for being with us today.
Thank you, Kelly. Thank you for the introduction. Let me just share my screen and you can tell me how that is going. Are we full screen now?
Yes, you're full screen.
Great, excellent. Well, I just want to thank you for the opportunity to address a topic that is something that I'm actually frequently asked about. Just to give a little bit of background of my experience in this space. I've been working in the biopesticide area now for actually 30 years. I started working in Novo Nordisk Entotech in Davis, California as a natural products organic chemist. And 5 years later, we started with a few people from that, handful of people from that company, a company called AgriQuest, where I also worked as a natural products chemist. And we operated in that arena over 17 years before we were purchased by Bayer Crop Science. And so we have been continuing on in this journey. And I've had a lot of different roles during that time, from bench chemist to overseeing chemistry and then discovery and eventually head of R&D at AgriQuest. And I also spent 10 years in field development, so designing and thinking about how best to run field trials for biological products, for microbial products. And now I have a role that is very much about technical communications and helping people, helping our growers, our customers, our own colleagues best understand microbial products, as they are quite different from other kinds of products that our crop protection colleagues are used to handling, and how they can be integrated into conventional programs, how they can be best used in best use practices for organic growers. So I have a large background in a lot of different areas. And one of the questions that I often get in a number of different collaborations I work on is, how do we actually get to product from where we're starting? Because there are many different areas where discovery is a key point, and that certainly has been the case with us as well. But also, what happens along the way after you kind of have that first discovery? And I think that this is something that I'm happy to be able to address. Just a kind of an additional background, one of the projects that I'm leading within Bayer has to do with citrus greening and looking at not only a microbial approach, but also a synthetic chemical approach, turning on plant host defense systems. And in that role, I've also interacted with a lot of different academic groups and helped on an advisory panel. And so this is another question that's come up. Once we actually find something, what happens next, and what's involved in that? So that's what I hope to be sharing with you today. And looking forward to some interactions at the end of this as well. So what we're seeing here is that, going from the idea or the discovery to the market, there are quite a few steps that we have to take into account. And I'm going to kind of use this slide that I've used to set up as a way that we're going to go through these different steps together and talk about what some of those activities are. And in this kind of overview slide, you can see that I'm addressing at the bottom not only biopesticides or biocontrol types of products, microbial products, but also biostimulants. And something when we think about development time and we look at different geographies, and I could see from the list that Isabella sent me that there are people on this call potentially that are from a lot of different countries and regions. And these kinds of steps can vary in different kinds of regions. So a little bit later, I have addressed some of what we see commonly in the US, but it's not the same in every country. So what you can see is development time also can change. So, if you're talking about biostimulants and we're in the US, it could be as short as maybe 4 years to get through a development process. But if we're going through a biopesticide process and we're in Europe or even the US, it could take longer than that. Obviously, it could be 6 years, it could be 8 years. It really depends a lot on the registration piece as well. But it's really one of the advantages we have is, even though that's a very long time, when you're thinking about these things, it's quite shorter than if you're doing a traditional crop protection synthetic compound. And the costs are also substantially lower, because one of the reasons is because of time, but also because of the kinds of studies that are required for this kind of process. And so what I'm going to walk through today is talk a little bit about this piece of discovery and research, and also the development part. And that's really pre-commercial. So I'm going to go through the different steps of what things we have to think about, what things we have to prepare for during discovery and research, and also during development, because at that point you would be either, if you're a company like Bayer, you're going to be commercializing it, which is additional cost in the tens of millions of dollars, depending on what geographies you're working on. But also if you are a university, for example, or a small company, and you're handing that to a commercial partner, this is what they will have to spend in order to move this kind of a product forward. So this is what we'll talk through today, and what are the kinds of things that we think about and what kinds of things we have to prepare for in order to move that kind of discovery forward. So, you know, sometimes it just starts with an idea, certainly with small companies, and I've been in a very small company where we were only 6 people in AgriQuest, all the way to a large company. But also, you know, the discovery is the very first step, and that could be identifying a strain, for example, or identifying a particular approach that you want to make. And then during this discovery and research, we're also thinking about characterizing how do we understand this strain or discovery that we're working on. Eventually, we will be thinking about strain optimization, and we'll be talking about these different kinds of processes, but that will lead also to field testing eventually as well. So let's see now. There we go. So we've got the discovery and research piece, and we're going to go through the different pieces that I mentioned, but we've also got development. So, you know, some of the discovery, we'll talk more about some of those specifics. Understanding biology during this discovery and research piece is something that we have found to be very important for helping our customers, our growers, our technical colleagues understand how use this kind of a product or product concept in the field. So what we have not found to be successful is we have something that looks really interesting in the laboratory, and we just kind of throw it over the fence to the people that are running trials. And in that situation, we have not had a lot of success. So the more we can put behind understanding what's happening with this product concept strain, the better we can help our colleagues understand how to have success when they're testing it. And we'll talk more about this as I go along today. Certainly for a biological microbial product, fermentation is a very important point, and we have to think a lot about understanding the fermentation process as well. And then we can move towards that development piece where we'll talk more about field testing, whether that's the early screening, whether it's registration trials, whether it's positioning trials, because what we want to do is put all of this learning and understanding towards actually commercialization, moving towards commercialization, where we're thinking about things like the regulatory, preparing for the regulatory process, making sure discoveries are protected, and even thinking about how are we going to actually produce this and make it available to customers. So, you know, at the top of our list, and very important for thinking about how a product will get through to the market, is thinking about safety. And this means that there are several different aspects we can look at. We can be actually running specific studies. We can understand something about the history or what's known in the literature about strains that we're working with and families of strains. And we have some powerful tools we can use to understand some of these different aspects. So I guess some of the questions we ask is, you know, we know that as we move further along this timeline, that we're going to spend more and more money on things like running field trials. So is it possible that we can use genomic information to help us understand the potential for what this strain might bring to the marketplace? Certainly when I started in this field, as I mentioned, that was actually in 1990, we didn't have access to these kinds of sophisticated genomic tools that we use today, and that many of the companies that are online with depend on heavily, including our own. And so, that's a really powerful way that we can understand potential of strains and also understand pitfalls of strains. And so thinking about what kinds of pathways might be in strains that we're looking to develop that might cause problems for a regulatory agency. Perhaps it's capable of making a particular compound that would not be acceptable for registration. This is a way to do some in silico research at a fraction of the cost, really, compared to moving a discovery or a strain so far forward in the process and understanding towards the end of that process that we have a problem that has to be addressed. So I think this is an aspect that we consider part of the safety portion of the research and development. And it's something that can really help with kind of cost savings. Another thing that I would say is more traditionally thought about as safety is what kinds of simple tests that are non-GLP can we carry out to assure ourselves that we are not exposing either our lab scientists or our field operators to any kinds of hazards that we're not aware of. And so one of the things you can also think about is before we're actually either scaling up processes or going out to the field, can we do some simple non-GLP tests like a simple mouse toxicity test or some other kind of indicator that would help us assure ourselves that we're moving forward with something that looks like it could eventually be commercial and that we're not, causing any kind of concern. There are even some countries like Germany as a country, for example, that requires some bee testing before they go outside to the field. So knowing that ahead of time to be able to kind of set up and prepare for getting some of that kind of information is really helpful. And one additional thing is that we want to make sure that whatever we're testing is pretty plant safe. We know that things can look different from the laboratory to the field, and so some of those kinds of early things that can be done can help in the long run, better understanding and better preparation for moving something forward on this journey. So the next thing we're going to talk a little bit about is efficacy. How well does this work? And of course, this is always top of mind for developing a product. And some of the things we're going to be doing in this kind of discovery and research piece is thinking about, first of all, we're often testing in a Petri plate. We want to think about how is this going to work on a plant? What kind of profiling can we do in the field? And so some of the kinds of things we want to consider is that, making this jump from the laboratory to the field is often a really big challenge. And when we're testing things in a Petri plate or a 96 well plate format, this is a great way to do discovery, and it's something we rely on, but it often doesn't represent the same kind of challenges as trying to control a pathogen on a plant in a system, or trying to get the same kind of uptake of nutrients in a case of a biostimulant that we expected to see from our in vitro type of work. So screening on plants, whether that's in a growth chamber or whether it's in a greenhouse setting, this is another really important step to help us prepare. And even in that situation, you don't have the same kinds of environmental conditions, you don't have the same kinds of light. And so once again, even from the growth chamber on plants or greenhouse to the field can also be a challenge. And so characterizing efficacy, this is something that's something we really have to think about and make sure that we're getting the best we can out of those early discoveries from the laboratory. Another thing that we have found to be very useful is to have the highest level of understanding we can about how a discovery lead is working. If we can help understand more about the mode of action, is it a foliar product? Is that what's causing the best efficacy in our greenhouse or growth chambers? Is it a contact activity? Is it something that actually needs to get close to the roots to colonize the roots? The more we understand about these kinds of things and what kinds of effects we might see from an environmental point of view, environmental conditions, that can help us in transferring that knowledge to the people that are going to be running things in the field so that they use the best practices. They're thinking about what are the right intervals, how do we develop the kind of information we need to actually have success in the field, and what do we do to give them that information to help them with that kind of process. So, thinking about the efficacy during the research phase with field trials, we're thinking about things like understanding what is the right dosing that we need. We really have difficulty trying to predict that from our greenhouse and growth chamber trials that we run. Frequently, we have to change the dosing once we get to the field. There's a lot of different kinds of conditions. The level of disease pressure or nutrient content in the soil could be quite different from what we're testing in our controlled conditions. And so, answering some of these questions that we can in the laboratory can help us figure out the targets we want to go for. What crop should we be testing? What would be the best to see results? And another aspect of this is thinking about how fast do we get results, because we're making decisions in the laboratory based on these kinds of field results. And so short crop cycle can help. We can maybe get multiple trials run in the same year even. But answering these kinds of dose questions, how often do we need to spray this kind of discovery? Do we get any help out of an adjuvant, for example, that can help penetrate or do something to help with the efficacy? And what do we want to compare that to in the trials that we're going to run? So these are, at this point, kind of proof of concept during our research phase. We might often be running a standalone treatment where we're just trying to understand what's the level of activity we see out of this discovery strain. And we may also want to start looking at where we could put it into kinds of programs, even while we're still doing the research, to understand, would this be a replacement for a contact product? Should it go in with the seed in furrow? And thinking about what will happen when we get to the regulatory trials as well. So another thing we have to think about, as we're moving along in this journey, is that we really have to think about how are we going to have the best possible quality for the product that we have, and how will this help also with the cost of goods? In other words, we're not in the pharmaceutical business, so the cost of our end product cannot be, hundreds of thousands of dollars a year, because a grower can't afford that. So we have to start thinking about how are we going to improve these processes, help our strain to be produced in a way that is actually affordable for a grower, that can fit into their farming practices. And, as an industry, we really want to make sure that there are quality products on the market that have some reproducibility, that have some stability. And so in this piece, when we're thinking about quality, this is where we have to start thinking about our strain, the process and the fermentation development, and how we're going to underline those processes and improve them so that we can get to something that could actually be affordable by the grower, and that he can incorporate into the types of practices and storage conditions that he's used to. So here, we're thinking about how do we develop a biological process? And we're going to have to develop this fermentation process, and the goal is really like, can we get to a cost-effective, robust process that could eventually be a commercial process for production? And there are different pieces to this puzzle. So, one is having the strain, which is, the discovery being as stable as it can be. So sometimes we need to do some kind of traditional methods for strain improvement to make sure that we can get a very reproducible fermentation out of that strain whether that's in a shake flask or in another situation. We have to also consider the fermentation aspect of this. There are many different outcomes from a microbial strain depending on how it's grown, what it's fed, what are the physical conditions of that fermentation. And so there's a lot of experimentation that needs to be done during this research and discovery phase around these kinds of things, like what does this microbe want to grow best? What are the best conditions, the temperature, the pH? And so these are in addition to the fact that we have to think about the engineering, like how are we going to actually scale this up? These are all interconnected, and we're having to think about these processes during this phase. When we are thinking about this scale up, we're usually starting first on a Petri plate where we're isolating things and moving to a shake flask, for example, or a tube. So very small scale while we're looking at understanding something that is going to potentially be a future product. But one of the challenges is actually scaling up. So it's not always easy when we're cooking to take a recipe that we know well and suddenly increase that amount by tenfold because we have a giant family reunion coming and we need to feed 20 people instead of 2 at home. That doesn't always work with recipes, and the same thing is true with the fermentation process. So we have to be able to kind of work at different scales along the way to make sure that we adjust the processes so that at the end, when we're getting towards a commercial piece, that we're able to actually have success with that. So starting with a shake flask, we're moving into a small fermenter, and this can start to give us a lot of information about things like what is the optimal temperature, agitation, pH, what kind of food does this microbe like? So there can be a lot of experimentation here. And then at each stage, as we're scaling up and we're moving towards a larger format, we're checking these conditions and can we optimize these conditions to continue to get the best possible product out of this. And so we're stepping through different types of fermentation equipment that allows us to understand, can we actually scale this? Can it be a commercial process that can be affordable for the grower? So we're going to end up at the end at a pilot production scale that's large enough so that we can predict that we would have success when we go to an actual commercial production facility. So these are important pieces of along this research and development piece for a microbial product. So, we're talking about some of the, fermentation pieces, the media, the actual seed that we use to start this process in these fermenters. And then we also have to think about what is important for the activity. And this goes back to the kind of mode of action pieces and making sure that we understand how important are the spores, for example, how important is some biological chemistry that may be present, and how, once we have that fermentation, do we actually maintain those pieces as we're concentrating it and trying to get it into a volume that's manageable for growers. So there's going to have to be some downstream processing that happens, like could be centrifugation or filtration, before we can actually formulate to try to help with the stability. And at that point, then we can think about either what kind of format do we want to have. Do we want to have a liquid product? Do we want to have a spray-dried granule? And so this process flow diagram helps us think about what kinds of things are important based on what we've learned in the research area. So, scaling up the process during development, making this jump, which is usually a 10 to 100-fold jump in the manufacturing process, to this 10 to 100-fold. That's usually a kind of jump that we need to make where we can, again, validate the specifications and think about reproducibility, because down the line, when we get towards a registration process, we're going to be asked to make 5 batches and show that they are similar and have that data available for our regulatory submission. So fermentation, again, we kind of talked about active ingredients, thinking about, spores, thinking about chemistry potentially. What are the processes that we're going to have to use in order to capture all of the important active ingredients? We know, working with biological microbial products, that they're complex. They have many different components that are important for the activity, and capturing all of those and being able to quantify those during the process is also important. So analytical assay development for looking at these different components helps guide and helps us understand how we're doing in this kind of movement towards what we need for a commercial process. And something that is very important for biological products also is formulating these in a way that can make them stable. The closer we can get to something that is like products that growers are used to using, as shelf stable as we can make it, as little special handling required, that helps us with uptake, because we already know that we have to convince a very conservative group of people that is our customers that microbial products add value to what they're doing. So thinking about what kinds of formulations they like to handle, but also things like, do we want this to eventually be used in organic certified farming? That really limits what kinds of formulation ingredients can be used. And so we have to consider those things up front as we're trying to prepare and think about the formulations that are going to be developed, and think again about, do we want a liquid product? Do we want a granule? How is that going to work eventually down the line for the grower? Another aspect that is highlighted on this overview slide is thinking about intellectual property from the beginning all the way through the process. I think that, considering the fact that whether you're going to develop this yourself or whether you're going to find a partner to develop something, that they're going to spend tens of millions of dollars in the commercialization process for a biological product. So even if you're a nonprofit agency trying to do this and provide this for the growers, you need to be able to make back at least that amount of money in order to break even. And, corporations aren't nonprofit, and so they need to make some kind of return on investment. And so if your discovery is not protected from the beginning, and there's no way to keep another company from quickly copying what's been done, then that is not going to incentivize a commercial partner to step in. I've had, a number of situations where I have companies, extension people, mostly actually from the research side, approaching us and saying, hey, we found out this really interesting compound that you can buy from Sigma is going to solve this problem for growers. Or maybe it's a byproduct of some kind of industrial process, agricultural process. And that sounds great. It's really exciting to know that there's something out there that's already a known entity and, could have immediate commercial use. The difficulty becomes that someone is going to have to take that compound, strain, whatever it is, and go through all of these processes in order to get something that you could commercially hand to a grower in a package. And that requires a regulatory process. It requires trials. It's expensive to do that. And so it has to be something, if you're going to get a partner in there, that has some kind of value that can help them maintain all of those costs that will take just to get it to commercial. So I know that academic and universities are very tied in now to intellectual property, but thinking about this along the way to make sure that nobody is presenting things in a conference that would endanger your ability to protect that. If you really want to go for a commercial product and that's your goal, you need to be thinking about this all along the way, because everyone has to be able to regain, at least recoup their costs, and additionally make some money on how they can get products to market for the growers. So that's just a fundamental part of what has to happen to interest a partner commercially in moving something forward. So if we think a little bit about the development phase and thinking about how are we going to get this prepared for registration. Another thing that I've had happen multiple times is people say to me in different research areas, we just need a registrant. We have this great discovery and we just need somebody to take it to EPA and get a registration. And it's a lot more complicated than that. And so that's just something to be aware of. And I know there's a number of companies that are on the line, so you're aware of this, but just thinking about what that process is good to know up front. I'm going to give examples with the US, which has one of the simplest possible registration processes for biologics that there is in the world. And part of this is because they have a regulatory agency that is separate and is considering microbials in the BPPD area. And so they're looking at microbials. There's also a biochemical pathway. So understanding whether your product's going to be a microbial or a purified biochemical is important to know. But at least having these different divisions within EPA helps because they have some knowledge experts on microbes, for example. And this can really change the data packages that they're asking for. This has been for some time that they've had this separate agency and started registering biopesticides in 1948 in the US and started kind of sponsoring workshops to understand better about how we're going to work with biorational pesticides. And eventually, in the 80s, they put out some testing guidelines for microbials in this subdivision M. And this is where they set up a tiered testing system. And this is also where we see some quite big differences in costs compared to a traditional crop protection compound. Let's see here. So we're thinking now about safety in terms of registration and setting up for a registration package. So, you often hear referred to the 6-pack that's necessary for a tier 1 study for a microbial product. And that covers the kinds of things here shown on this screen where we have acute oral toxicity, acute dermal toxicity, inhalation, thinking about getting studies on eye irritation, dermal irritation, and skin sensitization. And so these are the minimum that you need for the safety aspect from a human health perspective that will be necessary for a registration package. And these are certainly not as expensive as the long chronic tests that are required for synthetic chemistry, but they're still, have a pricey GLP price tag as well. So tier 1 is what we go for with microbials in order to afford what eventually the market can bear. But sometimes you could end up in a situation where tier 2 data is required. This happens if you see significant toxicity in the tier 1 studies. And for a lot of people, this would be the end of the line because then you're going to start getting into kind of subchronic toxicology and pathology situation, and these are very expensive studies. So this is just something to be aware of. You need to have a clean results from your 6-pack toxicology set in order to avoid any additional tier testing for a microbial product. In addition to thinking about human safety, they're also going to be interested, and you will need to have a lot of non-target testing done and environmental safety testing done. So that includes across the testing against birds, against fish, invertebrates, and non-target plants, potentially honeybees. That's always something that is very topical. And thinking about non-target insects. So this is an additional set of information that will have to be developed by a registrant in order to submit a package for getting a commercial product registered, at least in the US. And there are additional things that come up when you work in other countries. And different countries have different requirements. So that is also helpful to have somebody who is an expert in the regulatory processes of those countries you're interested in working in. So thinking about efficacy now that we're moving towards this development phase, Here we're thinking more about registration trials and positioning field trials. So what does that look like? That again depends on the country you're working in. So for example, in the US, trials are required, but they don't have to be submitted. They're expecting that you have efficacy data in field trials in the field at the federal level. However, at the state level, that's different. So for example, California requires you to submit efficacy trials. These are certainly not as onerous as trials that are required by regulatory agencies in other countries, but data is required. And if we're thinking about other countries, typically the registration trials require standalone treatments, for example, in the EU, at multiple doses and compared to a standard and looking at the efficacy. And there, that percent control, if you're talking about a biopesticide, differs by country to country as well. So there's a lot of challenges in working in a lot of geographies. In some places like Brazil, you simply have to be statistically significantly better than an untreated. In other countries, like some countries in Asia, you actually need a certain percentage of efficacy standalone. So understanding that is important in your partner or in your own company for moving outside of the US. Another really important point for commercialization is how are we going to actually use this in the field? Because a grower is not going to use 8 applications of the same product over and over for resistance management reasons and many other reasons. So commercially, how are we going to actually position this in a program? Are we trying to go early in the program to help with plant health? Are we trying to go at the end of the program to manage residues in a conventional program by reducing anything at the end? So these kinds of positioning trials are very helpful at this stage for understanding what's the commercial value and can we put it in a program as a tank mix, as a rotation. Again, these kinds of important aspects to understand during the development phase for registration. I mentioned and touched on already stability and formulation development and how important this is because we need to know that the way we handle things in the laboratory, and if we consider room temperature to be 72, that's not what happens in a warehouse where they're storing materials. It's not what happens in the back of the truck. And so thinking about temperature and storage of these products and how they're going to be treated in the field is important for us to understand. And so a lot of stability studies need to be run, and also thinking about what we measure when we're looking at stability. Are we looking just at the CFUs? Are we looking at the biological chemistries? How are we measuring the quality of that so that we can maintain that stability? Another really important part during the development process. So, what you can really see from this is that there are a lot of steps that have to happen after you find this discovery. And, partnering and or working together on your own plan for that needs to be incorporating all of these different aspects and thinking about that. It is very valuable to have a discovery or a strain that has a lot of promise. But there are many, many steps that we have to go through in order to actually make that a reality for the grower. And that's what we really want to do. We want to provide alternative products for growers that can help address their needs and also address the needs of sustainability and environmental concerns. So, the discovery is a really exciting piece, and we love that aspect of it, but we just have many, many more steps, and we need to think about this even early on. And the earlier that you're thinking about these things, and whether that's partnering or on your own organization, the better chance for success at actually reaching a commercial product. So I hope that, this has kind of given you a little bit of an overview of the many steps that happen after you have a discovery and after you've found that really exciting and interesting strain, and help kind of prepare things for future success so that we can actually move as many of these things towards the marketplace where we see this market growing and where we'd like to see agriculture moving. I'm happy to answer any questions I can on this presentation. Thanks for your attention.
Thank you very much, Denise. That was an excellent presentation of a very complex process, in fact. We do have a number of questions, and I will go to the topic you discussed there at the end about stability. How do you control the quality after production, and more importantly, after commercialization?
So how do you control the quality after production and after commercialization? So it's difficult to control unless you have very specific. I think the question is about what happens after it leaves your hands, because not what you did to help with the stability. It's difficult to do that. You don't know how people are going to treat it. So we kind of assume worst case scenario. And if it's the situation where your product needs to be refrigerated, you need to just give really clear instructions. We've talked about different kinds of approaches. Should we put some kind of, sensor? We've done stability studies where we sent our product to a commercial warehouse in another country and put a temperature sensitive sensor in there and watched what happened over the course of 2 years. It's really difficult to control. And so that's why putting effort into making it as robust as possible up front is an important aspect, or at least understanding what conditions it could be exposed to in the field. I hope that addressed the question. I'm not quite sure.
I think so, yes. One of the other questions that we have is, of course, as you know, product development requires a lot of resources in terms of people and money. Big companies have an advantage over the smaller ones, but you've actually had the experience of being on both sides of it. Do you see some strengths or advantages that smaller companies actually have in the development process?
Yeah, that's a great question. I think the advantages that small companies have is that they can be very nimble. So they can quickly kind of reiterate, they try something out, it doesn't work, they can go back and change it quickly. They're not subject to the same kinds of long-term planning and prioritization and things. They can also think very much about a niche market. They can develop a product for a market that is much smaller than a large company has to justify going through the whole process with. I think there are different kinds of models. You think about the pharma model where you have a lot of small companies doing discovery and then handing it off to somebody who can afford to do the actual commercialization and human trials and things like that. I think small companies have advantages in that they can quickly change course and they can quickly adjust things within their own prioritization schemes. So that's the beauty. And the downside is you don't have the resource of money to be able to run all the trials you want to do or do all of the kinds of things that actually get it there. So there's pluses and minuses. It's fun working in both environments.
It's great. Well, you've had an interesting career in that regard, so you actually got to see it from both sides. So, concerning studies for registration, would you consider a chemical or a biological product as adequate, as an adequate standard to compare to a biological product?
I think hopefully this is about field trials. I'm going to address it as if it's about field trials. Is that what it sounded like to you?
Yes.
Okay, so I think it's important to understand how well it's working against a standard that's in the same kind of class. When we work with, for example, a biopesticide that is a contact product, it's not systemic, I like to see in the trials designed that it's compared to a contact synthetic product, because that has the same kind of intervals and it has the same limitations in terms of coverage needed and things like that. Comparing it to the best systemic synthetic compound, to me, that's kind of useless because it's not going to perform like that. We don't have the same expectations for microbial products that we have for synthetic systemic chemistry. And so I also sometimes like to benchmark against another biological product because those have more similarity. So thinking about the trial design and thinking about pitting it against things where you're expecting the same kind of mode of action, I guess, or performance level, I think that's important. It can also help you think about how you can move into a conventional program replacing things like mancozeb or other contact products if your product's a contact product. So I guess addressing similarities in terms of positioning and mode of action for your standards is what I go for.
All right, great. So where do you get the strains from that you test? Do you isolate them from soil, plants, or similar environments? Do you collaborate with scientists from academia that maybe have culture collections? But where generally do you get them? And where, in your case, because of AgriQuest and then working with Bayer.
Yep, so all of the above. So, when we were little AgriQuest, we were isolating, we had our own isolation program going for 7 or 8 years. When we were purchased by Bayer, they had a strain collection. Some of our collection came from academic collaborations. Sometimes we purchased strain collections. And even today, we look at strains that come from collaborators from different universities, from different organizations. So, no company can find everything and do everything. And so the more collaborations we can have, the more we can work with people, the better. And we look at all of these things as ways that we can find the best kind of things for moving forward to the market.
All right. Well, can you speak to the differences of registering a biopesticide versus a biostimulant in the United States?
Okay, well, I'm not an expert on the regulatory side, but and I know that they are still developing what does that mean, a biostimulant, and exactly what is going to happen. What we saw happen in the past with biopesticides was we had to kind of have an industry standardization, and the Biopesticide Industry Alliance, and now called Bioproduct Industry Alliance, industry group was formed early on, and in order to help benchmark and put some kind of quality behind that. Now the biostimulants are being brought into that kind of process. So I'd actually have to get back to somebody specifically on exactly what the requirements are, because some of those things are still being developed in the US, and trying to separate differences between a biofertilizer and a biostimulant, and it's complex, and there's a lot of conversation going on in the EU about this. But if someone wants to send me an email, my email address is on there. I can get to the regulatory expert we have and address that question specifically for the US.
Yeah, this is an issue, particularly with regard to the biostimulants. It's an issue of great interest to the Phytobiomes Alliance. And in fact, we even have a regulatory working group trying to figure out how to make it better and easier for commercialization. So I would encourage anyone to reach out to us as well.
Yeah.
Is it a big difference? Ooh, the thing just, if you're in the whole process, when you're thinking about the whole process, if you're looking at a single microbe or a mixed, maybe a consortium?
In terms of everything? I mean,
yeah, in terms of the whole development process.
Yeah. Well, I think some things would be the same. I mean, you're going to have the same kinds of questions around field trials and efficacy and things like that. We develop single strain products so far. That's what we've done, and that's what my experience is in. And there are people on this call that have a lot more experience with multiple strains. And so I think that, I can't really address that. For us, it's more complex, thinking about how we can get a reproducible, stable product, but we work so far only with single strains. So I think that, some of the things would be similar or the same, and some will be very different. And I just don't have the experience to speak to that.
Certainly several companies are specializing in the microbial consortia. So the next question is, to what degree do large companies genetically optimize lead microbial strains to enhance field efficacy versus, say, fermentation ability, as you mentioned during your presentation?
Well, I don't know what most big companies do, but I would imagine that anybody who's working with microbes is doing, at least some kind of strain improvement, whether that be just looking within the population for the individuals that are capable of upregulating certain production of certain biological chemistries, or whether that's thinking about phage resistance or whatever. I mean, we use traditional kinds of similar to plant breeding techniques where we're going through populations and trying to enhance towards better efficacy. I think I would be surprised if there's any wild type strain that got developed without any kind of improvement on the market. There's a drive to try to get the cost of goods down. There's a drive to try to improve, you know, the production in a fermentation process, and that can really be enhanced by even just a very traditional method of strain improvement. There's other methods that can be used genetically, but there's also a lot of talk about what that means for Europe, for example. And so I think a lot of the traditional methods are used by anybody who's working in microbial product production.
All right, is there anything that you can share with regards to Bayer crop science activities targeting HLB?
Yes, I can share that because I actually lead that project in Bayer Crop Science. We have a project we've been working on for more than 3 years, which is funded by the Citrus Research and Development Foundation, which is a grower organization in Florida. They approached every industry, big industry, all the big ag chem players 6 or 7 years ago, asking them to work on HLB. It's a reasonably niche market for a big company. It doesn't meet that, you know, hundreds of millions of dollars of product, but we came to an agreement with them to have some FTEs funded within Bayer Crop Science to only work on this problem. And we have 2 methods that we're going after. The main project was to set up a screening process that could help us find something through screening in the laboratory that would be relevant in the field for this obligate pathogen that is in the circulatory system of the citrus tree and transmitted by an insect, the psyllid. And so we developed a screening platform using surrogate organisms, and we have the approach from a microbial side, looking for microbes that could produce compounds that directly kill HLB or reduce the titer and that are not used in human health. And we also have a synthetic group working in Lyon, France, in Bayer, that have had a program going looking at synthetic compounds that enhance the host response, the tree's response, an immune system response to fight off the disease. So we have both of these programs working. We have some leads that are being tested in the field. We work with multiple academic collaborators at University of Florida, Texas A&M, UC Davis, in order to try to move this discovery piece towards something that could be commercial. We hope to continue working on this for another 3 years into the future with additional grant funding that we're working on right now. And so we also look at discoveries from outside our discovery as well. So people can give us kinds of discoveries that we can put into our testing platform.
Great. We have time for one very quick question, if it can be answered quickly. What are the significant bottlenecks in the R&D or the commercialization process that typically eliminate your candidate strains? So how do you narrow down your pool?
Yeah.
And what are those bottlenecks?
Bottlenecks could be it's making compounds that will not allow it to be registered. Some kind, not all natural products are safe, obviously. Another one is we can't scale it up. We can't get it to make the jumps in the fermentation process development. We can't make it cheap enough, or we don't see consistent activity in the field. There are many different possible bottlenecks we see with this.
Great. Well, thank you very much, Denise. It's been a pleasure to have you, and during this presentation, I think has been very informative to everyone regarding the process. So again, would like to thank Denise Manker, from Bayer Crop Science, and hope that you will join us on our next Phytobiomes Alliance webinar. Thank you and have a great day.
Thank you.